Hello, and welcome today to today's lecture on germline gene therapy. We just finished up the lecture on somatic gene therapy. And remember, somatic gene therapy is basically going through and treating the body cells of an individual. Basically, somatic gene therapy is the focus of going through and treating a patient without any possibility of the changes or the manipulations or the things that we're doing being passed on to future generations. Now, what we're going to be doing with germline gene therapy is thinking about what is it that we're going to be doing that could potentially be changing the genetics of all of the descendants of an individual that we're working with. Okay, so with the idea of germline gene therapy, this is almost as if we're going through and making a transgenic organism. But in case this time we're making a transgenic human. So the idea is that, like what we did with transgenic mice, transgenic organisms in that way, mice is probably the best example. I think that's one we used a few lectures ago. We're going to do the equivalent of in vitro fertilization. And so we're going to take together egg and sperm. We're going to put them together in a laboratory setting, allow fertilization to occur. And then what we're going to do within, again, germline gene therapy, like when we make a transgenic mouse, is we're going to take and inject in our new DNA into that fertilized egg. And because it's in that fertilized egg, what's going to happen is as that fertilized egg goes through the process of development, start to divide, produce lots and lots of cells. Those cells start to differentiate, start to become specialized, start to develop into the infant. The infant will grow into kind of the child and then the adult. What's going to happen is that DNA that we inserted into the fertilized egg over here is going to be found in every cell within this individual. And so that DNA that we injected in is going to be in all of the cells, all of the body cells, but most importantly, it's going to be in those germ cells, in the cells that are going to be involved with producing the eggs in a woman or the sperm in a male. And so because of that, the DNA that we inserted here expressed in all of the cells, the body cells, as well as the germline cells, the cells producing the eggs and the sperm, this DNA, because it's going to be in the egg and the sperm, can be passed to future generations. And so again, when we're taking a look at the comparison, in somatic gene therapy, that what we talked about before, we're going to be going through and treating a small subset of the cells in the body. We're going to be going through dealing with non-germline cells, somatic cells, the body cells. We're going to go through, we're going to make a change in those cells. It may not be permanent because those cells may die, attacked by the immune system, or they stop producing what it is that they need to be doing. So we often have to do multiple rounds of somatic gene therapy. But the important thing is, because it is not getting into this new DNA that we're putting in the somatic gene therapy, it's not getting into the egg cells or the sperm cells. It's not going to be transmitted to the future generation. Now, in contrast, germline gene therapy over here is going to be present in the egg cells and in the sperm cells, and there, because of that, it can be heritable. It can be passed from the parent to the offspring, and that offspring's offspring, and the offspring's offspring's offspring, kind of going on kind of in perpetuity. It's going to be changing it for all future. And so because of this, with that germline therapy, when we're putting it into the fertilized egg, it's going to get into every cell within the body, and potentially within that single treatment, it's going to ideally cure that patient and potentially cure it for future generations to come. Now, if we take a look at this, within the United States, approximately 2% of the newborns are going to be born with some type of genetic disorder. There's about 2,000 to 3,000 inherited diseases, which are range from about 30,000 Americans with cystic fibrosis down to the ADA, the adenosine deaminase deficiency, one of those immune system deficiencies that we talked about with somatic gene therapy in the last lecture. That may affect only 40 to 50 people worldwide. And so some of these are somewhat prevalent. Some of these are going to be incredibly rare. So if we look at it, again, somatic gene therapy is going to use our vector, normally a virus, to insert the DNA into specific affected cells, and we're going to infect the cells. We're going to infect these cells with our virus particle, and as that virus particle goes in, it's going to take in that cell, and in doing so, cause that cell to produce whatever it is that needs to be repaired, produce the protein that needs to be repaired in that case. Germline gene therapy, very, very different approach. What we're going to be doing in most cases is either working with an egg cell or a fertilized egg in the process of in vitro fertilization. We're going to take our new piece of DNA and we're going to inject it into that egg or into that fertilized egg. And in doing so, it's going to be present. Our altered DNA, our corrected DNA, hopefully, is going to be present within all of the cells of that body, including the cells that are going to be involved with producing the eggs and the sperm when that individual is an adult. And so this is very different than what we're talking about with the existing process of in vitro fertilization, where parents may go through and in the normal process of in vitro fertilization produce, half dozen or a dozen embryos, but then they may screen those embryos for the presence of a genetic condition. If that individual, if that fertilized egg, if that embryo has that genetic condition, they may choose not to implant that and have that, go through the process of pregnancy and produce an infant at the end. But they may go through and they do the in vitro fertilization, they test it, and they find out that it does, that embryo doesn't have the mutation or doesn't have the genetic condition that they're concerned about. And so they're going to choose that child, or at least that embryo, to be implanted into the uterus and development. And so normally, it's being done to test for genetic condition. We'd also use it to test for gender as well. And to be honest, it probably occurs. Now, germline gene therapy, though, is this idea that we're going to take our corrected DNA, what we're referring to as new DNA over here, inserting it into that fertilized egg in a way that's going to be incorporated into the cell's nucleus, into the chromosome, and it's going to be expressed in all of the cells within the body. Now, the problem with this is, again, because we don't know exactly how it is that we're going to insert that DNA in, and again, this is prior to CRISPR, that method of really directed gene placement. We run the risk that as we correct one gene, we could end up causing something else to have a problem. We could insert our new gene into or nearby another existing gene that's going to be important. And in doing so, we could kind of disrupt its function. We could cause it to become a lethal genetic disorder, in which case the zygote, or the fertilized egg, the embryo won't develop. Or it could be something that doesn't appear until much later on, potentially in adulthood. And so, even though we may be fixing something, we could be causing a new problem that we're not sure of for a long time after we do this process. Now, the idea with CRISPR and some other techniques is that we go through and we actually do a targeted replacement. So in this case, we've got the light gray box here. Don't worry about the names. We've got a light gray box here, which is damage. We've got our corrected dark black box down here. So we got a defective gene and a replacement gene. We can go through with targeted replacement, and the process isn't all that important. But what we're going to do is we're going to cut out the damaged gene and graft in the corrected version of the gene. And in doing so, what we end up here is the chromosome, where we basically went in and completely replaced the damaged region with the correct region. And so if this works, this kind of minimizes the risk that we're going to go through and cause, additional problems down the line. And this is very theoretical, at least at this early stage, but potentially with CRISPR, it's something that we can go in and do going forward. Now, germline gene therapy and germline technology has been used for a number of years on transgenic animals. And so we can produce new breeds of animals that express specific genes. And so we can take a look over here. This is a normal mouse. This is a mouse over here, which is overexpressing growth hormone. And so if it's expressing the growth hormone, it's going to grow bigger and bigger and bigger. And growth hormone is one of the hormones that athletes sometimes take when they're trying to do performance enhancing drugs. Now, the thing with this is that you may go through this and do it a couple hundred times and result in lots of unsuccessful DNA transfers. It's going to be relatively rare that we're able to put it in, have it incorporated, have it being expressed in all the cells, and having it be expressed in a way that can be passed on to future generations. And when we're working with mice, that's possible. If we want to do 100 different baby mice and study them, that's relatively easy. But if we want to treat a human disease, you don't want to test out this technique on 100 babies in the hopes that one of the babies will have their genes corrected. So we need something that's going to be a little bit better than what we've got, at least up until this point. The other question is, if we're going to be doing this genetic modification of human beings, we're really on a slippery slope. And so, there may be something over here that's going to be really important to do. And so we're going to treat a lethal disease, and that's good. You want to kind of eliminate disease from society. You want to eliminate suffering. We'll talk about the ethical models associated with that. And so if we treat these ethical diseases, that may be pretty good. What about if you treat genetic conditions? I've got a genetic condition, for an example, that, causes, an individual to, have difficulty. Maybe they're not, as active as what they would like, or they can't get around as much as they like, or maybe they got, arthritis, or they got something else going on. Should we be treating those things? They're not lethal, but they're impacting on the quality of life. And I think for, some individuals, they're going to say, if we can minimize suffering, improve the quality of life, that's a good idea. Others may be saying, it's a great benefit to, correct a lethal disease and not have the person die, but there are risks with all of this. And, are the risks outweighing improving a quality of life for an individual? And so that's a little bit further down our slippery slope. And then finally, you've got people that may be wanting it for genetic enhancement. We go back to the previous slide, and it's a parent that wants to have a NFL linebacker in the family because they make lots of money, and that's the parent's retirement plan. And so what they're going to do is they're going to get their child to have increased growth hormone being present. And so they're going to be, a big, bulky, strong lineman that's, twice as big as everybody else on the team. And because of that, they've got this genetic enhancement, and they're going to be a better football player, and the family's going to get a house, retirement, and all that. We would probably say that genetic enhancement for those reasons probably, potentially, isn't something that should be permitted. But where do we draw the line? That's something that, scientists aren't going to be able to tell you. This is something that society is going to have to think about. So if we look at a couple examples of that, what genetic conditions should be treated? There's a genetic disorder known as sickle cell anemia. And I think we've talked about this. It has a single amino acid chain, a single amino acid within a protein for hemoglobin is changed because of a gene defect, a damaged gene. And so if we take a look at that, it impairs the ability of these blood cells with a patient that has sickle cell anemia to carry oxygen. And so in areas where they don't have a lot of oxygen being present, so if they're exercising or if they're a higher altitude with a lower concentration of oxygen in the environment, the cells go from this nice, healthy, kind of almost donut-like shape to this kind of sickle shape. And this sickle shape can kind of clump up in the blood vessels of the body. They can cause a lot of pain for the patients that are doing it. And it can also cause some severe problems kind of later on. And so we take a look at it. So single gene defect, blood cells, we can treat blood cells fairly easily. Should we go through and treat sickle cell anemia? It seems like a pretty good idea up here. But then when we learn a little bit more about it, what we realize is that sickle cell anemia, having one copy, one of those damaged regions, one of that kind of incorrect information. One copy is correct, one copy is incorrect. The copy, in patients that have a copy that's incorrect, that have an allele or a copy of that damaged information, they are resistant to malaria. And so we could go over here, treat sickle cell anemia, eliminate it, but then potentially mean these people now are going to be more vulnerable to the blood disease malaria. Malaria, you get an infection, you get a parasite in the blood cells, it kills off the blood cells, and it makes it very, very difficult. And a number of people die every year from malaria. And so is this something that we don't completely understand? If we don't completely understand it, is it something we should be working with? Is it something that we should think about fixing? So another example of this, should we fix mutations that we don't understand? We take a look at this one, CCR5 delta 32. Yeah, don't worry about the name. It's a deletion mutation. It affects about 10% of the people in Northern Europe descent, and it protects against the bubonic plague. That's pretty good. A lot of things going on in 2020, but not quite the plague yet, but it's still early. But this deletion also protects against smallpox and HIV. So potentially, if we go through and trying to change this expression, if we're going to go through and fix it so that normal people are going to be normally expressing the correct version of this, now they're going to be more susceptible to smallpox and HIV. And so we've got a situation here, again, if we fix something, we can cause a greater problem later on because we don't know it. So we've got this issue. We've got the technology that allows us to do this, but should we be doing it? Should we be using this, especially, making changes that are going to affect future generations? We can do informed consent, potentially, on somatic gene therapy, body cells. We talk to the patients, or we talk to the patient's parents if we're treating a newborn, and we can go through an informed consent process. But how do you do informed consent where, you're making a change and it's going to affect generations from now, and it's going to alter what their genotype is. It's going to alter what their genetics are. It's going to alter their characteristics. And they have no say on that because this is something that's occurring well before they're conceived and born. Now, if we take a look at kind of an example of this, and we're pulling back to CRISPR. Remember, CRISPR as that focused gene therapy or mechanism to go through and alter the DNA, correct the DNA in a very focused way. A Chinese scientist in 2018 reported the birth of genome-edited twin girls. Basically, this scientist in China went in and disabled the genetic pathway that HIV uses to infect cells. Basically went in and induced a mutation in CCR5 delta 32 to protect these children from HIV. And the reason for that is that they had a parent with HIV. So the idea is if we go through, induce a defect in that pathway, they're then going to be resistant to HIV. Now, this occurred, it was reported, and generated a huge outcry. Lots of condemnation, lots of international calls for regulations, kind of a moratorium on doing germline editing. Just huge, huge issues. A lot of controversy associated with it. This individual is no longer practicing because of the outcry of this. He's doing some other things now that you can research, and they're interesting in and of themselves as well. Some ethical issues associated with that too. But somebody went through within the last couple of years, used this technology and did germline gene therapy and potentially altered at least these twin girls and their progeny, their offspring going forward in perpetuity. Now, soon after that, it became clear that a Russian scientist used the same way to edit genes in donated eggs. It allowed him to work with an HIV-positive mother that didn't want to, that didn't respond to the medication and wanted to have an HIV-negative baby. And so you could go through and do the same type of thing that the Chinese scientist did. He in theory, he's able to go through and work with a deaf couple that has genetic hearing loss and correct the genes, correct the DNA in their baby so that their baby is not going to have this. And this scientist, Denis Rybakov, says that he's done this. He's been able to do the editing. He's got them in donated eggs, but he hasn't done anything with them yet. He says, as of October 2019, he says he will follow internationally agreed upon regulations for this work. He's going to do it on, the embryos, but he's not going to implant them until there are better guidelines. There are no rules. There are no regulations internationally, or in many cases, in some countries, even locally, on the restrictions for this. And so, as I've said throughout this course, science, and I'm a scientist, so I'm a little biased, science is going to, develop new technologies. Science develops new techniques that we can use in a whole variety of exciting ways. But should we be using it? Should we be doing this? I could argue very strongly that creating transgenic mice as better models for the study of disease is a wonderful advancement in our understanding of how human disease works. But I don't know the implications, future implications of going through and making germline changes, making some of these changes like this Russian scientist or the Chinese scientist on the slide before. There's just a whole lot of unknown. There's a lot that we don't know. And until we do know what should be allowed and what should be restricted. And again, scientists can't tell you that. Scientists can tell you what's possible, and it's dependent upon others, including an informed populace, an informed government that can go through and determine what it is that should be allowed and what it is that should be restricted. So that finishes up our view of germline gene therapy. A couple articles associated with that, kind of related to the CRISPR, a little bit on the China crisis. Last year, NIH director calling for a moratorium on germline gene therapy. And then finally, a question, is this even ethical? Is this something that we should be considering? So I encourage you to take a look at these readings, come back for the last mini lecture where we'll talk about the activity for this course. As always, if you have any questions, feel free to email me. Thank you and have a great day.